sometimes the students almost get forgotten and aren't nearly as included, and so I really structured this seminar in such a way to ensure that students, I'm really targeting it towards students, and that is to say that at the end, please, students in the room, be thinking about interesting, provocative questions that you want to ask of the work, and please don't be shy when there are things that I talk about that are not familiar to you and you want to know more, or you want to push it to the next level of inquiry. So today's talk, this is a collaboration with obviously folks here at GW, folks at the Faroese hospital system, at the University of Southern Denmark, and also at the Harvard T.H. Chan School of Public Health.
It's probably on the WebEx. You have to just click on the screen once.
Oh, okay. We seem to be advancing this way. Do this.
Okay, that's all you have to do.
Yes? Great. So today I'm going to talk to you, first I want to tell you a bit about global sperm declines and what the literature is showing that we have seen very discernible changes and declines in overall sperm indicators, both in the U.S. and globally. So I'll tell you about those data. I wanted to talk to you about legacy chemicals, specifically organochlorines and DDT that are in our environment and that we are exposed to regularly, and they're very hard to get out of the environment. And so I'll tell you a bit about how they came into the environment in the first place and how we're exposed to them and how they're in our bodies. I want to tell you about the Faroe Islands, which is the setting for the research that I'll be presenting today, what's distinctive about it, and why was this an important place to study these connections between sperm and chemicals. I'll also talk about sperm abnormalities, and especially aneuploidy, which is an area of research that my laboratory has been focused on for a number of years. Mainly because it allows us to think even beyond simple indicators of sperm health into other important areas of fertility and reproduction. And then I'll tell you about the findings from the Faroes study, and I'll tell you about why it matters. You know, what does this all mean? What are some of the implications? So first, these trends demonstrate what's been happening in some key areas of male reproduction: testicular cancer, hypostasis, and sperm counts. And what we have been seeing over time, over the past approximately 50 years, are increases in testicular cancer. In hypostasis, this is actually an abnormality of the penis. It's the urethral opening. It's in the wrong place on the penis. And then these are sperm counts, mainly count, that's million per milliliter of semen. This is also a graph showing breast cancer, age adjusted. That would be for male and female combined, and what's been happening with breast cancer. Now, these data were first produced, especially related to testicular cancer, hypospadias, and sperm counts, focusing on the U.S. and then expanding to other parts of Europe, and then more recently they've been updated globally. So they were some early indicators that something was happening with respect to male reproductive health. And more recently, we had a meta-analysis that was able to bring together data across different continents, and that includes North America, Europe, Asia, and Africa. So again, 20 years ago, the data were suggested primarily for North America and for Europe, and then more recently, researchers have been able to bring together data for Asia and Africa. And what you see very distinctively are significant trends of decline, about 50% decline from where we were 50 years ago in terms of overall sperm counts. So, very simply, men are producing about 50% less of the sperm that they were producing 50 years ago, and that is true across multiple continents. These observations pertaining to testis cancer, sperm counts, and and then congenital abnormalities of the penis and the testis have been brought together under this framework of a testicular dysgenesis syndrome to explain a discernible pattern of decline in male reproduction. And really one of the key researchers that put forward this idea, hypothesized this idea, is Skkakebaek from Copenhagen. And he pointed out that we were observing a number of characteristics: cryptorchidism, which is undescended testis, decreased testosterone production, impaired spermatogenesis, and also testicular cancer. And so he brought us back to where might this be happening? And the hypothesis is that it is related to, in part, environmental exposures. Certainly genetic defects and polymorphisms that would predispose to these problems are having a role. Other lifestyle factors that impact sperm health certainly have a role, and the potential epigenetic factors, most of which we really have not even begun to identify, but undeniably would have a role as well. And it's thought that some of the key points of time in which this would have effect is during the fetal germ cell development or fetal Leydig cells, and I'll tell you a little bit more about those types of cells. So these exposures impacting testicular dysgenesis, resulting in ultimately these conditions, health conditions, causing reduced male fecundity and influencing pregnancy rates. So this is about, I'd say, 30 years of research coming together in the current state of thinking. Enter environmental chemicals. So you saw here, this is one of the quadrants, and that would be environmental exposures. And I want to talk to you about some environmental chemicals. For those of you that are new to your program, I'm sure you'll learn more about organochlorines. I don't have time to go into all of the details and chemistry of the compounds, but suffice it to say that they are persistent organic pollutants. They're in our environment. They're considered legacy chemicals. They originated, PCBs originated from for their insulation properties, and they have discernible characteristics that were great for producing building and construction materials, which included everywhere from window casings to insulating foams to insulating materials that would go inside of containers. And the problem being is that they last in the environment for a very, very long time. Their half-life is in the frame of decades. So they persist even though they've been banned for quite some time, since the 1970s. The same is true for DDT. So DDT was originally developed in the 1940s. It was seen as a very promising insecticide. It was used to first control insects, but then it started to be used for agricultural purposes in a more broad, widespread way, until we discovered that it was having impacts on our wildlife. And in the US, it was banned in the 1970s. And in reality, both of these types of chemicals continue to persist in our environment. This is a picture from Tanzania, and to this day, DDT is being used for vector-borne control and indoor residual spraying. So DDT is still very much in use in various parts of the world, specifically to combat malaria-carrying mosquitoes. Whereas PCBs are banned, they're no longer produced, but they linger in our environment. And when I say linger, these are data from the National Health and Nutrition Examination Survey. So the U.S. does periodic assessments of what are the chemicals in our body, and they sample a representative general population sample of Americans, and then they test for various chemicals. And they've tested for over 200 chemicals. All of these data are easily online through the CDC. So if you have a chemical that you're really interested in, and it's pretty well known and has been established, chances are the NHANES study has evaluated that. So here are data for specific types of PCBs and also the main metabolite of DDT. And so here are the years that the NHANES has been measuring these chemicals. So 1999 through 2000, 2003 through 2004. Here are the number of people in which they have actually been able to measure these chemicals. So I want you to know about this as a data repository, really useful. You can look at phthalates, you can look at other types of pesticides, you can look at metals. So these are some PCB and PBE data. And I just wanted to point out, you can follow geometric means. So for PCB 118, just one of the many types of what we call congeners, congeners. The level is 6. The unit is nanograms per gram of lipid, which is in serum, in blood. And then here are some other PCBs, and here's DDT, or para-DDE. And you can see some evidence of the levels going down, but at the same time, they are certainly found, and they're found for, well, let's say 50th percentile. 50% of the population would have a level of 203 nanograms per gram. So they are common in the average person's body. It's not as though you must have had to have been working in agriculture in order to be exposed to DDT or have it in your body at any one point in time. So they're still ubiquitous in our environment. Okay, so that's the PCBs and DDT. Now we're going to move to the Faroe Islands. They are a little known cluster of islands that are north of Scotland, halfway between Scotland and Iceland. They're 18 island archipelago, and the population is 50,000 people. And they have been studied, this community, they are a Denmark protectorate, but they have their own self-governance, their own governmental structure. And this is the series of 18 islands. They're all connected by tunnel, if you believe it. So you don't have to take a boat to get to the different places, but they can drive via tunnels in order to get to the different islands. They're a really beautiful place, as you can imagine, quite lovely. It's a very calm, serene way of life. Obviously, most people know each other at the 50,000 people. Lots and lots and lots of sheep. And the reason that they're of interest for environmental health studies is because they have relied for thousands of years on sustaining themselves via fish. So their diet is composed largely of fish, and in particular whale. And they hail from the tradition of the Vikings, and so they have been consuming whale blubber for hundreds and hundreds of years. And what has come to be understood in the last 25, 30 years is that these whales, on top of the food chain in many ways, are filled with organic environmental contaminants. And it means that it's making it into the bodies of the Faroese population. So over the course of 25, 30 years, there have been studies, many of which have been done by my colleagues who are collaborating on this study, understanding how those exposures over time, exposures, for example, to mercury and heavy metals, exposures to organochlorines and other pesticides, dioxins, have influenced the overall health of the residents of the Faroe Islands. And so this is an image that is not uncommon in the Faroes each year. They have an annual whaling event where the residents of the island, mainly men, the fishers, will go out to capture hundreds of pilot whales. These are not endangered species. There's an abundance of pilot whales. And so they will go ahead, they will hunt them, they'll capture them, they'll bring them in, they'll push them into shore, they'll slaughter them on the shore, and then start to divvy up the meat. And so generations have grown up doing this, and it means that children get a lot of exposure to pilot whale blubber. And there's a great movie documentary that I think we'll bring to the department and have a showing. It's called The Island and the Whales. And it is a very even-handed exploration into this practice and recognizing what the chemical impacts are and the health impacts are of relying on a diet heavy in whale blubber. So, this was an important setting for us to be able to study how chemical exposure to persistent pollutants, for example, would be impacting reproduction. And the big question that we wanted to ask is, there had been a number of studies demonstrating these organochlorines impacting overall sperm health. We were most interested in this part right here. This is an illustration of when sperm cells originate and what the process is. And we were most interested in abnormalities that are manifest in chromosomal aberrations during meiosis. So in the testis, got another picture here. In the testis, this is a cross-section of the testis. There are different types of cells that are going through change all the time. It's a very dynamic action. Women are born with all of the oocytes that they'll ever produce. Men are producing them on a daily basis. So there are multiple sperm types, sperm cell types at any one time. This is an example of what the cell division process is from spermatogonium, which has two forms of the chromosome. It first divides also into meiosis 1, which still has two, and then into the secondary spermatocyte, haploid, second meiotic division, spermatokids, and then sperm cells that are haploid, one copy of the chromosome. I know this is in the weeds, but that is to say that the prior data had shown associations between high levels of organochlorines and sperm concentration, the number of sperm in a sample, the motility, how the sperm move, and also morphology, how they look, their overall shape and size. But no studies had looked at these other indicators, including sperm chromosomal abnormalities. And so this is just a reminder. I'm not sure if everyone studied meiosis anymore. When I was in high school, we studied meiosis. I'm not sure if folks are still studying meiosis, but this is a reminder of how meiosis happens, which is the exchange of sister chromatids, such that the chromosome itself has 4 unique parts, and it happens in a very well orchestrated way. It's actually traveling along the meiotic spindle. The separation, the exchange happens, or the recombination happens via the microtubules. So there's an orchestration and an architecture to meiosis, and the big question is it possible that testis that is influenced by chemicals that are biologically active, they're actually manufactured to be biologically active. DDT was produced so it would be effective in killing things. A chemical that is known to have hormone disrupting properties, can that interfere with this well orchestrated, finely honed type of process of cell division that's very hormone dependent? And it relates to a much larger question of the way in which chemicals known to have hormone properties or that can disrupt the process, they're referred to as endocrine disruptors, whether or not they're influencing the normal reproductive processes that are happening all the time. The same question has to do with metabolic processes, immunologic processes, etc. How do exogenous chemicals influence the normal process? And so this is why my lab has been delving more deeply into meiosis, because we certainly believe that meiosis is hormone dependent. So, process during sister chromatid exchange, things can go wrong, and the accurate exchange number of chromosomes may not happen. And when that occurs, it's called aneuploidy, which is an abnormal number of chromosomes. And it could be that the chromosomes did not cross over, or it could be that an extra chromosome crossed over. So one of the two can happen during meiosis. And when that happens, it results in discernible impacts. So it occurs in about 5% of clinically recognized pregnancies. It is thought to account for at least 1/3 of all spontaneous abortions. It's aneuploidy in the sperm or the egg is thought to be responsible for even upwards of 80% of spontaneous abortions are just babies, the fetus not carrying to term. And it's responsible for a large portion of infertility, miscarriages, spontaneous abortions, infant death, malformations, mental retardation, and behavioral abnormalities. So aneuploidy is a big deal, both in the egg and in the sperm. And so here's just a review. This is from a mouse model of what happens. So this sperm should have just one sex chromosome, not two. Remember we said that it was haploid, it should have just one. And it has an extra. So it's fertilizing an egg. This is the oocyte fertilizing the egg. After 1 day old of fertilization, you've got two X chromosomes. Then as the embryo grows, it's going into the second cell stage, day and a half. You have two cells. Both of them have an extra X chromosome. Now the 4-day-old embryo is complete with an extra X chromosome. So all cells in the embryo carry an extra X. Okay, is that important? The reality is when it comes to the sex chromosomes, the X and the Y, it results in a variety of what we call viable conditions. So that sperm is still capable of fertilizing an egg. Many other autosomal or somatic chromosomal abnormalities result right away in an inability to fertilize. But for reasons unknown, the sex chromosomes, if there's an extra in an egg or a sperm, they're still viable. And they result in offspring with discernible conditions. So there's Klinefelter, that's an extra X. It occurs in between 1 in 500 to 1 in 1,000. Some of the characteristics are hypogonadism and sterility. Turner, it's missing a chromosome, an X or a Y. 1 in 2,000. Jacobs, that's an extra Y, 1 in 1,000. And then triple X, 1 in 1,000. And this tells you what is thought to be the paternal source. What % of the cases are attributable to a problem with the sperm? And so 50%, 80% for Turner, 100 because it's always a Y coming from the male sperm, and then just 6% for that extra X. And each of these, we would say, phenotypes are of consequence, everywhere from infertility to developmental delays to intellectual problems in intellectual development. So when I first started doing this work, a lot of times when I would present, people would say, okay, so you're trying to link this sperm abnormality with this chemical exposure. We know chemicals have been on the rise. You can look at most charts that would demonstrate where we were, not for DDT and PCB in terms of productions, but many other chemicals have been on the rise. And so what about aneuploidy? And the short answer is it's a real challenge to answer that question, especially for the sex chromosomes, those conditions that I just talked to you about, because oftentimes those conditions are not diagnosed at birth. You can't tell. You can't tell when a baby has an extra X or is missing. It's usually manifest at adolescence. So number one. Number two, we don't do routine karyotyping of babies in the US. So there's no registry to register when in fact you discover that someone has an extra X or extra Y or is missing a sex chromosome. So this is an example of data from Denmark to demonstrate when was it most likely that Klinefelter, that extra X chromosome condition, was diagnosed. And you can see it's in the 30s and 40s. So we don't have good data for how prevalent is Klinefelter's or Turner's or triple X syndrome, certainly not in the US. But there are data to suggest that from Europe, I won't show you those, but to suggest that they have been on the rise in the last 30 years or so. Very limited data, but the studies that have looked at a meta-analysis suggest that Klinefelter's has been on the rise. Okay, so the big question we wanted to ask here, we've been studying this for a while. We've looked at organochlorines and we've seen the relationships with adult aneuploidy, organochlorines measured in adults. And we wanted to see if we could find any evidence of aneuploidy being a higher incidence among men who were exposed as babies in utero. And so this is the study design that we looked at. So we're evaluating fetal germ cells in embryonal exposure during the fetal period. Our prior studies only could speculate whether or not the chemical exposures happen in utero, but this study we were able to look at it more carefully. So the design is from the Faroe Islands cohort study. We first had a random sample of about 100 men. These men were first entered into the study about 30 years ago as they were in the womb, and their mothers were evaluated. So the moms were entered, and then their offspring were evaluated over time. The big question had to do with the influences of mercury in the overall intellectual and cognitive development of the babies. Was in utero exposure to mercury predictive of where children would be at age 7 or at age 14 in terms of intellectual development? So we had men that actually had been in the study for the entire 30 years of their life. And so we could take their semen at adulthood and we could look back and what was their levels of organochlorines in cord blood. And then we had a much smaller group of simply adult men where we had semen and serum, and then we had another group of adult men who had children, and we could evaluate their semen and serum. So sampling from each of these sources, the one really, really important, most difficult to get would be the men for which we had their cord blood levels and then their adult serum levels and their semen levels. So that's the design of the analysis. And I wanted to tell you about the way that we look at aneuploidy. We identify the chromosomes of main interest, which are the sex chromosomes, X and Y, and we are able to use DNA probes to focus in on the DNA of the centromere that indicates whether or not the chromosome is there. And the challenge of this work, which I didn't know when I first took it on, I said, okay, we looked at semen quality, concentration, morphology, but motility, but what about aneuploidy? But I didn't understand how rare the condition is and how, an abnormal sperm cell would be, and that therefore you have to look at thousands upon thousands in order to get a true indicator of whether or not you have changes in the frequency of aneuploidy. So we have to do spot counting, which is to evaluate thousands of sperm cells and looking at the chromosome presence in every single one of those sperm cells. So we're looking for X or Y, or we call it the autosomal control 18. So imagine scanning a man's sample for as many cells as you can and really being able to count exactly how many had an X, how many had a Y, how many were missing one of them, how many had an extra. That's what those are our data. So it requires, when I first started the work, all of the work that had been done in this area before had been manually, and I pledged that I was not going to subject graduate assistants to that kind of drudgery, being in windowless dark rooms and just shifting through the microscope different filters in order to, by hand, count how many cells there were. I promised we were going to do that, so for years now we've been working on a semi-automated system. It started in my work at Harvard and then continued here, and I'd say that in the last 2 years it's really ramped up and been optimized and performing much better. So it allows us to accurately detect how many of the thousands of sperm cells that we look at have an extra chromosome. So our analyses, just wanted you to know what our methods are for evaluating differences across the exposure groups. We use a Poisson regression, generalized estimating equations, and that is because the outcome aneuploidy frequency really is a count, and it varies according to how many nuclei have you stored. So Poisson allows us to utilize the aneuploidy frequency as a count variable rather than as a dichotomous variable. And then we've looked at the extra X, the extra Y, either one, X, Y, and also total as our outcomes. And then the exposure variables are the most prevalent, the four most prevalent PCBs. I showed you that list of different PCB congeners. So we've looked at the most prevalent, we summed them, and also PPDDE, which is the most common metabolite of DDT. It takes a while for DDT to break down into PPDDE, and it would follow that because exposures haven't been new, they certainly can be continuous but not new. By the time you're measuring it, most of it has been broken down into the metabolite PPDDE. And then the variables that we're concerned might be related to both the exposure and the outcome. Those would satisfy the criteria. The confounding variable would be age. So the older you are, for example, the higher levels of exposure you might have, and age is susceptible, sperm aneuploidy is susceptible to age as well. So we want to hold age constant. Absence time, when was the last time that the man ejaculated? That influences how many sperm cells you have, how fresh they are. Could certainly influence your ability to get a decent sample for analyzing aneuploidy. Smoking, we know, is can influence aneuploidy risk, and it's thought that it can sometimes influence chemical exposure, including hand-to-mouth contact. And then the other semen parameters we often keep in just for good measure to make sure that the relationships that we're seeing aren't driven entirely by the overall quality of the sperm, but rather really a chromosomal abnormality. So those are our confounding variables that we've used many times and that we include in the model. Okay, so here's the men for which we had all of those data that I mentioned. 100 men. Their average age was 23. Their BMI was 25. The concentration we measured by, again, million per mil was, or this was the percent that was less than 15 million per mil, so low concentration was 15%. So that's to say that there were 15% of the men that had low concentration. The others were average, were normal, above 15. Motility, less than 40% was the 5% of the sample, so only 5% had poor motility. 44% had greater than 4% normal morphology. Morphology is probably the weakest indicator of sperm health. It's hard to score, it's very subjective. It varies across different laboratories. It's not uncommon for, that's why it's set at such a low level, that only 4% or more or less would be considered abnormal. Absence time is the number of days that they had ejaculated prior to us receiving the sample. We always ask them to abstain from ejaculation for 3 days, but it varies. So this is the absence time, and then 42% were current smokers, so a pretty high percentage of the population were current smokers in the Faroes. So remember I told you that one of the big challenges of doing sperm aneuploidy research is that you have to get big samples in order to come up with an accurate assessment of how many are abnormal. So these are the number of sperm cells, the mean number of sperm cells that we scored, 8,500. This is the median, the 75th percentile is 10,000. These are the conditions that we looked at, XX, YY, XY, and then total. These numbers should be saying to you, this is a really rare event. It's less than 1% that what we would see there would be an aneuploid condition. Less than 1% of the sperm that we scored, there would be an aneuploid condition. So the overall total was 1.82 when we summed these three together. Rare event, but as we've pointed out many times, the consequence of which is very clear and exact and has some considerable consequence. And I said that our system has been performing better. 10,000 sperm, 8,000 the mean. That's much better than we've been doing, let's say, 5 years ago, which may have been around 3,000. So we're really happy about our ability to go into higher and higher numbers of cells to get even more accurate record of aneuploidy. And then these are the levels of DDE and PCB exposure in cord blood and also in the adults, some of which we didn't have the full number of measures for, so that's why it's a reduced N. And this is microgram. What I showed you before, I think, this is also, nanograms is what I showed you before in Haynes. This is micrograms. And so 0.92, which would translate into about 92 micrograms cord blood of DDE, about 98, nanograms of cord blood of the sum PCB, 0.31 or 31 for the adult DDE, and 45 for the adult PCBs. So you can see that cord blood was much higher. So that was measured back. If the men were approximately 30 years old, and more in their late 20s, early 30s, it was measured back 30 years before. So we took a sample when they were in utero, and then we took a sample at the time that we took the semen. So it's about almost a third the level here. Yeah, just about a third decrease over the course of 25 to 30 years. Okay, so here are our findings. This is for adult DDE exposure, and we look at it according to tertiles. So we take the full distribution of the men, we break it, chop it into 3 even parts. This is the incidence rate ratio, the incidence of aneuploidy for these conditions. So again, remember we're studying an extra X, an extra Y, an extra X or a Y, and then these 3 combined is total disomy. So these are adults, and we saw that there was a significant increase from the lowest for DDE to the highest. That was for XX, for YY, also for XY, and for total disomy. So across the board, adult DDE levels were associated with an increased risk of each of these chromosomal abnormalities, and that was significant for DDE. And that was also true for the main PCBs as well. Now, these findings we have reported before. It was what we found with a larger sample. We went into about 150, originally we reported on 100. Went into 150 men and found very comparable patterns for the adult levels. But again, I told you that we were intrigued by the notion of fetal exposure being the most determining, the most deterministic of what's happening 30 years later. And from an epidemiology perspective, that's a really difficult design to accomplish. You can do that with mice, you can do it with rats, you can breed and raise, you can dose with intention, and you can evaluate what kind of impact it's going to have on the fetus in utero. But with adults, it's very difficult. So I think we're intrigued by the idea there might be an association, but I have to be honest with you to say I never believed there would have been one. I didn't think that we would have found this. It seemed far-fetched. It seemed, theoretically interesting, scientifically interesting, but I didn't think that we're going to find it. So here are the data, and this is, again, DDE cord blood, 25, 30 years before, and these are all significant increases, and even more so than the adult levels were. So remember, the adult levels were 30%, or about a third less, or a third of what the cord blood levels were. So we saw pretty strong associations and stronger associations, including these incidence rate ratios are higher than they were for the adult levels, and then for total disomy. So this is DDE, and then this is for PCBs as well. So it was almost a discernible dose response. And the folks in my lab are here. They can attest to what my reaction was when we got to this point in the data. When I first saw it, it wasn't an aha moment. It wasn't an aha moment. It was a, oh no. It was an oh no moment, because it basically said the most important point in time when these influences, exposure influences are happening is in utero. And I just didn't expect that. And I presented this to the folks in the Faroes in June, and you know, scientists can get really caught up in their findings and kind of forget the public health message. And I was somewhat hesitant to say, Paul, you know what I found? I found that the in utero exposure was much more strongly associated with the risk for sperm aneuploidy than the adult levels were. So that was the main finding. We're working on writing up the paper. And here are some of the other papers. I mentioned that, so we reported the adult exposures before and said we saw a significant association. This is in Boston men. This is a sample of 192 men coming into a fertility clinic. Exactly the same study design, just with adults. We took their serum, we took their semen, we looked at whether or not men with higher levels of PCBs or DDE had a higher risk of sperm aneuploidy. So that was actually one of our first studies that we did in Boston men. So we did find that. We've also found suggestions of associations with contemporary use. So these are legacy chemicals, but our contemporary use chemicals, including pyrethroids and organophosphates, we've found some associations with the risk for aneuploidy as well. And that's why we refer to it as a cross-sectional study. Same point in time, we take both types of samples and then look at associations. So it's cross-sectional, whereas the cord blood study was, in many ways, prospective. So the implications are that for PCBs, we saw approximately for adults an increased risk, or the adult level PCB exposure, an increased risk between 10 and 22%, and for prenatal exposures between 17 and 66%, and then for DDE, 32 and 52% for adult, and 3 to 75% increase for prenatal. So I wouldn't call that good news necessarily.
Melissa, can you just clarify, what's that? That is a percent increase above what?
No increase. No increase. Above 1. 1 would be no increase.
Right, but is that above the, whatever you're finding, the 1% risk that you usually find there? And
yes.
Is this going, so is this saying it's going up to 23, or is this going to 1, or going to 1.5%? Or?
It's basically an odds. It's an odds. So it's not as though we can predict that 75% of the men with this exposure will develop increased aneuploidy. It's the men with this high level of exposure have a 75% higher odds or incidence of sperm aneuploidy.
Okay, so, but it's the incidence, okay.
It's a probability.
Okay.
So they're at higher risk.
They are at higher risk, and it's a measure of magnitude.
Okay.
It's a measure, it's a
Can you talk about
ratio.
the range and the percentage, especially for the DDE for 3 to 75% of it is?
It depended on the condition. So for the XX, it was less. YY was very sensitive. Increased risk for YY as a type of aneuploid condition is why you have that range here. So we just sort of aggregated it and said that range somewhere between. So I'm invoking the words, the somewhat cynical words of a colleague at Boston University, an environmental epidemiologist, who has been quoted over the years as saying that what's the definition of a public health catastrophe? A health effect so large even an epidemiologic study can detect it. Because, as you know, we really struggle with, we can't prove causation, and we really struggle with even interpreting what our increased rate ratios, incidence ratios, and odds ratios are saying about causation and the impact on the population. And I often, through many grant submissions, have a lot of experience from skeptical reviewers about, does this really matter? Okay, so it's a rare, first of all, it's rare that you're going to have, there are millions of sperm. You just need one to conceive. It's less than 1% that might be abnormal. And then the probability of a aneuploid offspring is also relatively rare, somewhere between 1 in 500 to 1 in 2,000. So does this really matter? But those were the same arguments that were put forward 25 years ago when it came to air pollution. And those small changes in risk for respiratory disease, for example, from air pollution were seen as, is this really that significant? The same argument as it related to subtle changes in IQ score. A 0.5% difference or 0.5% difference or 1% difference on the IQ test, is that really important? And it really is when you think about it from a population perspective. So do I think that organochlorines are the cause of infertility? No. There are many factors that would relate to infertility. But do I think that the presence of environmental exposures could be affecting the normal process of spermatogenesis and impairing chromosome division and increasing non-disjunction, along with other processes, spermatogenesis processes? Our data continue to converge to suggest yes, and would explain in part those declines in male reproductive function. So I think that's what this is saying, is that do it again and again and you find the same thing. That's when you start to say, there's probably something here. And then why does it matter? PCBs, they're banned, they're no longer produced. DDT, okay, we stopped producing it, we've solved that problem, it's no longer an issue for this country. But the reality is, number one, in vector-borne disease control, DDT is still the preferred chemical and is seen as the most viable option. And interestingly enough, I plucked this out of the New York Times article from a couple of years ago. When Zika emerged, DDT all of a sudden started to be discussed again. And so we need to bring into the larger discussion chemical options. What are the long-term impacts on the choices that we make? And so I'll leave you with this. I'm not saying that I get all of my scientific information from Gentleman's Quarterly, but this is from an article in Gentleman's Quarterly. And it talks about, it's very interesting reading, it's just from this past month. It talks a lot about global declines in sperm, what this means in terms of our fertilizing capacity and our ability to replace our civilization. And it conveniently shows these two graphs. One is global productions of plastics, millions of metric tons, and then the overall decline in sperm count by the millions. Now, I would never encourage my highly discerning, discriminating students to make any kind of conclusions about this except interesting coincidence. But I did like the quote, and that is, the chemical revolution gave us some wonderful things. New medicines, new food sources, faster and cheaper mass production of all sorts of necessary products. It also gave us a living experiment on the human body with absolutely no forethought to the result. With that, I want to thank the many hardworking, bright minds in my laboratory and other collaborators that contributed to this work, and the funding sources as well. And I welcome your questions, especially first from the students in the audience that maybe learned something more.
Thanks for your presentation.